carboxyl group somewhere, right? We have a carboxyl group somewhere. Carboxylate ion and hydrogen ion. So this is how it forms. Like they release a hydrogen ion and they convert it into an anion. Similarly, you have another ionizable group which is amine group, NH4 plus, NH3 plus, H plus, right? So there is two ionizable groups in amino acid. Okay, first is your carboxyl group and second is your amine group. Okay, and the carboxyl group will be protonated if they are present in low pH. Low pH means highly acidic, right? Highly acidic means they must have released H plus. Okay, high pH means basic, right? Anyone who can tell me range scale of pH?
0 to 7 acidic and then from 7 to 14 basic.
Okay,
Something.
so it's below 7 it's acidic and after 7 it's basic. Okay, if it's acidic there must be a protonation from carboxyl group. If it's basic there must be a protonation from amino group. Okay, or amine group. So, and this is how you have to calculate pKa based upon how these two amino acids are different. Okay, they comes with different structures, but Dr. Nayan told he might do some calculations and discuss about this later on. So let me see how long I can go. Okay, I think you might know what is the cation and what is the anion, right? Positively charged ions are cations and negatively charged ions are anions. And in a molecule, if you have both positively and negatively charged ions, we call them as Zwitter ion. Okay? So the best example to understand Zwitter ion is this structure. Can you see this? They have a positive charge and they have a negative charge. But this molecule is electrically neutral. By electrically neutral, I mean they overall do not have any charge. They are neutral, but in molecule itself they have one positive and one negative charge which cancels it. Okay? So amino acid with both positive and negative charge are called as Zwitter ion. Okay, it could be a subjective question in your paper, right? The Zwitter ion, define as Zwitter ion. Okay? So a molecule having both positive and negative charge in the same molecule and they are electrically neutral are called Zwitter ions. Okay? So they both have positive and negative charge, single molecule which is just the same on each side. And this is how a pH and OH equivalent graph looks like. Okay? And this is called PI. PI means isoelectric point. Okay? So let's understand these three terms. PI, which is isoelectric point, Zwitter ion, cation and anion. Okay? So PI is isoelectric point. So isoelectric point is the value of pH. Okay? Isoelectric point is the value of pH where Zwitter ion is present. Okay? Did you get that? Isoelectric point is a pH value where Zwitter ion is present. It means where amino acids are electrically neutral and they have a positive and negative charge at the same time. Right? You see this? This is how you understand this graph. And there are two pKa values, pKa1 and pKa2. Right? Just a few minutes ago I said amino acid has two ionizable groups. One is carboxyl group, another is amino group. Okay? Carboxyl group releases their hydrogen at low pH and this is called pKa1. Okay? Or pK1. The another which is released in high pH is called pK2. And Pi is average of pK1 and pK2. So you can write Pi equals pK1 plus pK2 divided by 2. So your Pi value is average of 2 pK value. One in the basic side, other is acidic side. Okay? Did you get that? So that's why you see this. They are present in their cationic form when they are at their lower pH. Okay? and the higher pH they have a pK2 value which has present in carboxyl form. Okay, and this is a Zwitter ion which represents the value of pH where they are electrically neutral. Okay, and this is 5.97 in that case. In this case, okay, it could be different depending upon how you draw the structures and how R groups are different in each amino acid. Okay, and there's a whole lot of calculations you might see or want to do some calculations or few numericals. Same as I said, Pi is equals pK1 plus pK2 divided by pK divided by 2, right? So let's calculate Pi value. If I give you a pK1 as 3.12, write down pK1 equals 3.12 and pK2 value is 8.9. Tell me what is Pi?
6.01.
6.01. How? Add divide by 2. Make an average. Okay? Can you do that? So depending upon R group it varies. Okay? And it's a busy. That will take a whole class. That's why I'm not opening that thing. Okay? So that's it. pK1, pK2 divided by 2. Let's see how two amino acids are connected to each other, which is called polymerization, and this is a process where there's a removal of water. Okay? Can you see this? You have to now recognize amino acids. This is an amino acid. You see that? This is carbon, no carbon. This is special carbon. We call this as alpha carbon. Okay? And there is another alpha carbon. You should be able to recognize alpha carbon whenever you see structures like this. Okay? This is alpha carbon attached carboxyl group, NH2, NH3 group, R group, and this is a hydrogen. Okay? Ideally it should be somewhere here, right? Another amino acid with another R group, R1 and R2, it represents two different R group. It could be anything. It could be alanine, tryptophan, phenylalanine, or anything else. Okay? So hydrogen goes from NH2 and hydroxyl group goes from carboxyl group, and there is a removal of water and they form amide bond. The same amount you are asking about, right? CONH. Okay, and this is amide bond, and this is more famously known as peptide bond. Okay, write down this is a peptide bond. Okay, and when multiple amino acids form a chain like this is called polypeptide. Okay, you understand that? A polypeptide is polymerization of amino acid, and removal of water happens at its tip. Okay, so polypeptide. So how many peptide bonds are present in a polymer of two amino acids? One. So imagine if I ask you a question, how many peptides bond are present in amino, like a polypeptide of 100 amino acid? 50? 99. So it's n minus 1. Okay? So imagine if you have another amino acid over here. Okay? Another CO, OH, hydrogen, there will be two peptide bond. 3 amino acid, 2 peptide bond. 4 amino acid, 3 peptide bond. 50 amino acid, 49 peptide bond. Okay? So another tricky question. Okay? Let's move ahead and see how peptide ends are not the same. Why? Because you started from here, right? See, carbon, and then another carbon, another carbon, another carbon, another alpha carbon. Okay, everything is alpha carbon. But you have a amine group at this terminal, but you have a carboxyl group at the other terminal. Okay, that's why this is called as amino terminal of the amino acid, polypeptide, and this is called carboxyl terminal of the amino acid. In short, very famous. This is called N terminal, and this is called C terminal. Okay, so imagine if you have a polypeptide, 100 of amino acid. Which one you call as first amino acid? The one which starts with nitrogen. Okay, So this is first amino acid, second amino acid, third, fourth, fifth. 5 amino acids. Okay, this is called pentapeptide. Okay, not a famous name, but number of amino acids, and you have different R groups over there, right? First is, let's brush up your knowledge, and let's see how much you remember. The first R group is anyone? Serine, CH2OH. If it's a S in place of oxygen, cysteine. The second is glycine. Third one, tyrosine. If I remove this OH, phenylalanine, right? This one is leucine, right? Isoleucine would be this CH3 over here. And if I remove this CH2, valine, right? B. Right? You should not be, you have to opposite this because it's already flipped, right? This is B, means valine. This is leucine, right? I think you'll somehow remember, right? Need 3 more practice and that's it. Naming of polypeptide. Different ways to remember. S for serine, G for glycine, Y for tyrosine, A for alanine, and L for leucine. Next time when we'll meet, I'll ask you the questions. Because not everything is like S and S, G and G. Look this. This is tyrosine but represented by Y. Okay? And they are tryptophan and other amino acids. They are named differently. Okay? So you have to remember those names. Next time we'll meet, I'll ask you the questions how to, use 1-letter code or 3-letter code. Okay? So yeah, that's it. This is full name of amino acids, serine, glycine, and then you have to, figure out what are what. Okay? Okay. So let's end up here. Dr. Nayan will go forward and teach you more about the purification method. And to here we are different variety of functions. There could be a hormone, pheromones, neuropeptides. There could be antibiotic protection or useful protection like amanitin, which is present in mushroom. This is a very famous poison. Anyone? Amanitin. They are present in mushrooms. They are called deadly mushrooms. So mushrooms could be poisonous. The most poisonous things is mushroom. They have a very deadly toxin inside them, like amanitin. But this is for their protection, okay? Not to kill us from the animals mostly. Could be polypeptide, coenzymes, or prosthetic group. I think there are other amino acids which are present in body, but they are in modified form. By modified, I mean tyrosine is also a modified amino acid, right? It's a modified amino acid, like naturally. But our body also modifies some of the amino acids in their way, okay? Like selenocysteine. Now they included this as 22nd amino acid, okay? 22nd. Can you see the difference? CH2OH is serine, right? CH2OH is serine. CH2SH is cysteine, and CH2SEH is selenocysteine. Seleno means selenium. Okay, so nature is smart. They are modifying things and making a new amino acid. So cysteine with a selenium is selenocysteine. Can you see this? 4-hydroxyproline. This is a proline, but in 4th position they have a hydroxyl group. That's why they are called as 4-hydroxyproline. And this is 5-hydroxylysine, 6 and methyl lysine means 1, 2, 3, 4, 5th methyl lysine, right? Gamma carboxyl glutamate. Okay, this is alpha, beta, gamma carboxyl. You see that? Naming is logical. Okay, if you understand the logic, you don't need to remember the name. Okay. My God, this looks horrible. Okay, good luck. Then selenocysteine and pyrrolysine. This is a pyrrole group. Okay. This is how you remember the name of the amino acids. And this is some of the more modified amino acids we have. Okay. Next, purification and other things will be taught by Dr. Narayan and we'll see you again, and all the best.